Good afternoon viewers and welcome to the EduSat lecture for the science stream. Today we have with us Dr. Charu Dogra Rawat from Ramjas College Department of Zoology from University of Delhi. She has been teaching you many aspects of molecular biology and has explored the fine world of DNA. We all have DNA or some sort of genetic material. The bacteria has it, the fungi has it, the plants, even the animals have it. So what are those differences that separate a virus from a human? We will look at those aspects of DNA, its structure and its conformation, and we will try to learn more about the impact of conformation on the functioning of an organism. So over to Dr. Charu Dogra Rawat.
Thank you, Unnati. A very good afternoon to all the listeners. And as Unnati said, we'll continue with the saga of molecular biology. Today I'll be talking about the genome organization, and we'll cover up the prokaryotes as well as in our subsequent lectures about the eukaryotes also. So we talked about the structure of DNA, what the DNA is made up of, but now how it is present in the cell, how it is packaged to lie inside the cell or the nucleus, as we talk about in the case of eukaryotic system. So we'll look at that and we'll start with the prokaryotic system today. So the learning objectives for my today's lecture would be, first we will understand the concept of chromosome, like DNA, how it becomes a chromosome or how it is basically packed up inside the nucleus or the cell. We'll understand the genome organization in bacteria. As I said, we'll be talking about the prokaryotic system first. So we'll understand how this DNA, how this genome is packaged inside bacteria. You will have an understanding of the genome organization in viruses. The most important viruses or the most model organisms which we use is basically the Tobamovirus virus, and as well as we'll talk about the genome organization in the T4 Bacteriophage. We'll also understand the genome organization. We'll start with understanding the basic genome organization in case of eukaryotic systems, and we'll look at when the cell is lying at the interphase stage of the cell cycle, when it is not dividing, in fact it is preparing for division, then how the genome is organized inside it. And we'll understand about the concept of the two terms which you must have heard, eukaryochromatin and the heterochromatin. So we'll start about talking about the chromosome. So what is a chromosome? Within the cell, the DNA is associated with proteins. So DNA, as we talked about, is a polynucleotide chain. It has got a sugar, a base and a phosphate, but this DNA double helical structure is associated with a lot of proteins inside the cell. Each DNA molecule with its associated protein is called as a chromosome. Now what does this chromosome serve? This chromosome serves several important functions. Why this organization, or does this genome organization has any significance in the cell? Yes, it does serve several important functions. The first of which is that it helps the DNA molecule to fit inside the cell. Now you know that the DNA, the cell is very small. So the DNA, the entire length of DNA has to be incorporated inside the cell. So this fitting is possible because of its association with the proteins and the organization of DNA into different levels of structure. The second important function which it serves is it protects the DNA from the damage. So the naked DNA without the proteins is more susceptible to damage by radiations, by heat or by any other physical factors. Association with the proteins renders it or protects the DNA from these kind of damages. Then another function is that it is transmitted efficiently to both daughter cells when a cell divides. The inherent property of DNA is to pass on the information, the hereditary information from one generation to another. So the DNA present inside a cell when the cell divides has to be transmitted into the daughter cells, and this transmission is efficiently done if the loose DNA molecules are organized into well-defined chromosomes. So if they are defined into chromosome, the transmission becomes very efficient. Then the overall organization of DNA regulates the gene expression as well as the recombination between parental chromosomes that generate diversity. So the regulation of gene expression is an important aspect. The DNA is not just lying as a linear or a circular molecule, but it is organized into higher order structures which tends to bring different parts of DNA probably in the vicinity of each other, tends to bring the regulatory elements nearer to the genes, which basically expresses into proteins that gives the cell a particular function. So this kind of overall organization renders the gene expression to be regulated, as well as the recombination. The most important part in creating the diversity amongst the organisms is also the recombination which occurs when the cell is dividing. So this recombination which occurs during the meiosis is basically between the parental chromosomes, and this overall organization into the chromosomes helps it a lot. So that is why the DNA is organized into a chromosome. These are the functions it serves under the cell, and it is very important to understand this genomic organization inside the cell. So we will start with the prokaryotic system, and we will see how this DNA is organized inside the bacteria. The DNA or the entire genome of a prokaryotic system is termed as nucleoid. The chromosome of, for example, E. coli is nearly 1000-fold compacted in a nucleoprotein complex called the nucleoid to fit approximately 1/5 of the cell's volume. So the length of DNA molecule is very large, but the volume of the cell is very small. Since it's a prokaryotic system, it does not have a well-defined nucleus. So since it does not have a well-defined nucleus, the chromosome lies inside the cell. Now inside the cell, it is folded approximately 1000-fold compacted because of the presence of the or association of the DNA with the proteins. The bacterial genome sizes vary between less than 200 kb to over 13 MB, and cell sizes ranges from 0.2 to 750 micrometers. This inevitably entails very different degrees of nucleoid condensation. So because the length of DNA varies as well as the cell volume varies, different kind of compaction can occur in the DNA. But the baseline is that the DNA does get associated with proteins and condensed together to be fitted inside the cell. This nucleate compaction, the nucleoid compaction, is achieved by a couple of factors. The one is that the cytosol has a high density of macromolecules and by the effect of excluded volume. So there are a lot of macromolecules which are present inside the cell, and that affects the nucleoid compaction. Then the DNA is negatively supercoiled by topoisomerases. We talked about the DNA topology in our previous lecture, that the DNA is not lying flat on the surface, but it is coiled over its own axis, or the twists are there within the DNA double helical molecule, which gives it a kind of a supercoiling. And we also talked about that the DNA both in prokaryotes and eukaryotes are negatively supercoiled. The specialized enzymes which induces these negative supercoiling are called as topoisomerases. So this negative supercoiling ensures that the nucleoid is more compact in nature. And then the condensation by the attachment of nucleoid structure proteins. Every cell has certain structure proteins. We will see down the lane that what these structural maintenance proteins are. So every cell has these structural proteins which are associated with the DNA and leads to the condensation of the DNA or the compaction of the nucleoid present inside the bacteria. One of the models which is prevalent about how this genome is organized inside the bacteria is the Rosette model. The first investigations into the structure of the bacterial nucleoid came in the form of light microscopy of stained specimens. For example, certain stains are like Feulgen and Giemsa, by which you can stain the DNA, and electron microscopy of thin sections. The nucleoid is mostly separate from the rest of the cytoplasm, and it is not bound by a nuclear membrane. The essence of prokaryote being a prokaryote is that it has got a primitive nucleus. So basically there is no nuclear membrane, and the genome is lying, or the DNA is lying on the cytoplasm only. So it appears to be separate from the rest of the cytoplasm. Some DNA strands extrude from the nucleoid into the cytoplasm like loose pieces in an otherwise tight ball of yarn. So you can see in the figure that there is a central tight ball of yarn, and then there are basically different loops which are, or different strands which are extruding out from this yarn. It can be the supercoiled domain, or there can be a scaffold which is present, which is holding the yarn in the center, and then there are 100 or 50 kb independent loops or domains For example, just 12 are shown here. In this electron micrograph, you can see that there is a central core and there are many threads which are protruding out from it. So these DNA strands extrude into the cytoplasm. It is not a very compact. In the center there is a core, but rest of them there is a protrusions which are lying outside in the cytoplasm. So nucleoid is located in the center. It is structured into small supercoiled loops that are relaxed independently when DNA is interrupted. So when the DNA has to replicate or when the DNA has to transcribe, then this supercoiling is basically done away with, and this supercoiling is relaxed so that the two strands can separate and the transcription or replication can take place. Otherwise, all these strands which protrude out from the central compact core is in the negatively supercoiled form. These domains of relaxation protect the chromosome from breaks that would otherwise lead to cell death by total loss of supercoiling. So we talked about that when we were talking about DNA topology, then the supercoiling relaxation has to be localized. So there are certain domains which allows, these protrusions allows the easily relaxation of the negatively supercoiled DNA for the processes such as transcription and replication. If it would have been a very compact and condensed yarn or ball of yarn or very compact central core only, then when it has to uncoil, then there was more chances of nicks or breaks to be produced. That is basically the damage in the DNA can occur, and which will be really devastating for the DNA, for the existence of the DNA. So these relaxation domains protects the chromosome from breaks and makes it easier for the supercoiling to be relaxed so that the processes such as essential processes such as replication and transcription can occur. So now if we look at the elements of the genome organization, while bacterial genomes are immensely fluid in terms of gene repertoires. What does that mean? Now DNA has genes basically which are defined. The genes are the segments of the DNA that can be transcribed into RNA and can give rise to a particular protein that can carry out the function inside the cell. So these genes, there is a whole lot of variety of genes which are present on the bacterial genomes. However, they are extremely conservative in the terms of chromosome organization. So the organization of DNA is practically very constant or very conserved. However, the type of gene can vary. So you can see in the figure that you know that when the replication is there, then usually there is a leading strand and there is a lagging strand. Then the replication starts at, for example, the ori, the origin of replication, and these are all showing the different parts or different segments of the DNA which are present inside the genome present on the genome of the bacteria. So there are highly expressed gene clusters at ori for replication associated gene dosage effects. There can be a gene strand bias, results in more genes in the leading strand. So these are certain characteristic features of a genome organization. The functionally in bacteria. The functionally neighbor genes are co-transcribed in operons. A very important concept in case of prokaryotic system is that a multiple set of genes involved in a common pathway are put under the control of a common regulatory material. We will see when we will talk about the transcription regulation, but there the multiple genes whose proteins are involved in a common type of a pathway, for example, say the metabolism of lactose, all those genes are present side by side and are present in tandem and put under the control of a common regulatory element. And such a structural unit which is formed is called as an operon. So the genome organization, the functionally neighbor genes are co-transcribed in operons which group again, then multiple operons can be grouped together to form the superoperons. There are genomic islands which are often the result of the lateral transfer, which basically means that a common characteristic feature in case of bacteria is that the DNA can be transferred in horizontal manner, which is basically not from the parent to the offspring, but the multiple bacteria lying under the same niche has the possibility to pass on the DNA from one to another species, which can be by conjugation, transformation, transduction, any of the methods, but the DNA can also transfer from one bacteria to another, and that way they can incorporate the segments of DNA from other species within its genome. So that give rise to what are called as the genomic islands. So genomic islands are very common in case of the bacterial genome. Then the leading strand COPS motifs near differential guide chromosome translocation. For this transformation location, there are certain motifs which are present, can be found on the strands. Then replication terminates at the when you talk about the replication, we know about these things. Then there are certain macro domains which are the superstructures of supercoiling domains. So we talked about the supercoiling domains. Together these supercoiling domains can form certain macro domains. The recombination associated chi sites are there which are accumulated in the leading strand. So chi sites are the sites specific where the recombination occurs. Then there are replicores which have similar sizes, that is chromosomes are symmetric. So if you see, then right hand side and the left hand side, the chromosomes are symmetric in nature. So one such unit is called as a replicore. Then there can be a bias which is GC skews are higher in the leading strand. So when A is equal to T and G is equal to C is not there, and one of the strands has a high concentration of G and T or C, G and C, then that term is called as the GC skews. So that bias is also found in the leading strand. So such is the organization. So organization is very constant. Now what kind of genes are present on it can be very diverse on different species of the bacteria. So this is about the genome organization we are talking about. In the next slide you can also look at the scales of the genome organization. So when we talk about in the terms of length of the DNA molecule, then initially there are motifs which are very small regions, you know, the supercoiling motifs, the regions of DNA. Then supercoiling motifs together can give rise to certain repeats, then repeats and motifs can give rise to genes, then multiple genes put under a common regulatory control can form operons, then multiple operons can give rise to certain bigger domains. These domains, then the islands which we talked about, they are bigger in size, then the domains and islands and all these things together can give rise to macro domains, and finally there are replicores. For example, in the case of a circular DNA molecule, there are two replicores which are present. So that is the scale of genomic organization in case of bacteria. So now we talk about the genome organization in viruses. So we take the example of two viruses. One is the Tobamovirus virus and another is a T4 bacteriophage, a virus that infects the bacteria. That is a bacteriophage. So these two viruses we study a lot about their biology in molecular biology. So we have taken, we'll try to understand the genomic organization in these two viruses. Now as you know, the viruses genomes can be single stranded or a double stranded molecule. It can be a linear molecule, a circular molecule, or in fact a segmented molecule also. The single stranded virus genomes can be a positive sense, that is of the same polarity, the nucleotide sequence as the mRNA, or a negative sense. So this is how we describe the single stranded virus genomes, because the single stranded virus genome will replicate, will transcribe into the mRNA. If it transcribes into a RNA and then that it first replicates into the double strands and then the other strand transcribes into an mRNA, then that is said to be the positive sense, because the strand of the DNA, the single strand of the DNA is of the same polarity, which is 5' to 3' as is in the case of the mRNA. A negative sense is the opposite of it, and sometimes the single stranded virus genomes can be ambisense, that is a mixture of the two sense can be present, that some part of the DNA can be in the positive sense and some part of the DNA can be the negative sense. Then virus genomes range in size from 3500 nucleotides to approximately 1.2 million base pairs, again a very large diversity. And unlike cell genomes, virus genomes may, that is the important part, that it may consist either of DNA or a RNA. Usually the bacterial chromosomes or the eukaryotic chromosomes are DNA in nature, but in this case it can be a DNA or it can be a RNA, and we have talked about the structure of both these nucleotide molecules. So this is the diversity of the genomes which are present in the viruses. So we'll take up first, the virus genomes may contain information that can be recognized and decoded by the particular type of host cell. Now you know that virus genomes contain this genetic information, but mainly it depends upon the host cell genetic information to make more copies of itself. So the virus genomes should have at least essential genes which are present there, so that they can be recognized and decoded by the particular type of host cell. So we'll start with the tobacco mosaic virus. We'll look at like it belongs to the genus Tobamovirus. There are 15 members of Tobamovirus which are there. They are naked, that is they are usually not associated with any proteins. They are rigid rods, plus it is a single stranded RNA which is unsegmented. So there is one molecule of RNA which is present. This RNA is single stranded RNA and it is unsegmented, so not divided into any parts. No family assignation by ICTV. So this council which basically assigns the different families under a genus. So no family is assigned to the tobacco mosaic virus, it just belongs to the Tobamovirus genus. So that's how it looks like. It looks like the rigid rods which is shown on the left hand side, and on the right hand side there is a zoom up of one of the rods, and you can see that there are 2130 protein subunits approximately which are present. Each subunit is associated with 3 nucleotides. So we will see that how now this DNA is organized inside the tobacco mosaic virus. So there is a protein capsid which is present outside, and the single stranded RNA molecule which is helical in structure protruding out from the units which you can see is there. The 49 nucleotides and 16 1/3 protein subunits per turn. So there is a helical single stranded RNA molecule. So approximately 49 nucleotides and 16 1/3 protein subunits are present per turn, and there are 130 helix turns of protein subunits. So approximately 130 helix turns are present inside this whole DNA molecule. So these are some of the numbers which we look at when we talk about the genome of the tobacco mosaic virus. Now if you look at the genome organization, the genomic RNA is only 6.4 kb. So it's a 6.4 kb transcript which is present, and this picture is basically telling you that the genome organization is as such that there are only 4 genes which are basically there. You can see there are different colors, the dark blue color, the brown, the yellow and the light blue color. There is a 5' end, and on the top of it there is a cap because it's an RNA molecule. So there is a cap which is present, and then there is a non-translated region. On the right hand side, which is at the 3' end, there is a tRNA like structure which is present in the genome. Now you can also see that the first transcript is approximately 126, or a protein which is formed from the transcript is 126 kilodalton protein, and another protein including the same part of the RNA molecule forms which is called as the 183 kilodaltons. So there is an arrow which shows that there is a leaky stop codon. So there is an amber codon which is present here. When this amber codon is active, that is the translation just stops there, then a 126 kilodalton protein is formed which is involved in the translation of the genome. Then there is also a 183 kilodalton protein that can be formed if this stop codon is surpassed, and this protein is basically a replication associated protein. Then there are certain subgenomic promoters which are present. The yellow part is basically for the transcription from RNA negative, that the RNA which is negative in the sense then the transcription occurs and you get a small part of the mRNA which is there. As I told you, then it has to be transcribed again, it has to be become double stranded and then has to be transcribed. So you get another transcript, and this transcript can translate into a 30 kilodalton protein which is basically involved for the movement of the particle. Then there is a small blue part which is again transcribed to form and then translated to form a 17 kilodalton protein which is basically the coat protein. So it basically gives rise to the genome of a tobacco mosaic virus, give rise to only 4 kinds of proteins, one which is involved in the translation, one which is associated with the replication, one which is for the movement of the particle, and another which is basically the coat protein. So this is how the organization of the genome in the tobacco mosaic virus looks like. So similarly, you can see here that there is a 5-methylated cap, but there is no 3' poly-A sequences as you see in the case of higher eukaryotes or in another prokaryotes. The 3' poly-A sequences are not there, but there is a tRNA like structure which is present at the 3' end. The expression is similar to that of toga viruses, another family of viruses producing non-structural proteins by direct translation of the open reading frame encoded in the 5' of the genome. So, as I told you in the previous slide that there is a open ending frame, so direct translation occurs. So, there is no transcription from that part, the direct translation occurs into two different types of proteins. One is a 126 kilodalton, and because of the presence of this leaky stop codon, it can be 183 kilodalton protein. And then in the 5' of the genome, and the virus coat protein and non-structural proteins from two subgenomic RNAs. So, we have got two subgenomic promoters which were localized here. So, these two subgenomic promoters transcribes the rest of the part of the genome, the yellow part and the small right hand side blue part, which transcribes into two transcripts, and then they gave rise to two proteins, one which is a 30 kilodalton protein involved in the movement of the particle, and another is a 17 kilodalton protein which forms the coat protein of the tobacco mosaic virus. So, this essentially is very important, and the important part is about this leaky stop codon, which is the information which is used, which is asked a lot of times. So, that is how the genome is organized in the tobacco mosaic virus. If you just look at, just to go a little deeper, because I thought till this much is enough to know about the tobacco mosaic virus, is how this RNA molecule is assembled, because you can assemble the virus particles in vitro. So, how the RNA molecule or the self assembly of the tobacco mosaic virus takes place. So, the figure shows the stages in this self assembly. So, the first is that the intercalation of the initiation region of RNA into a disc of capsid monomer. So, capsid is the outer protein coat, and they are formed, they are basically polymers. So, a lot of protein units, subunits come together to form these capsid units. So, the RNA, which is single stranded RNA, and some they are associated together also. So, some portion of RNA called as the initiation region of the RNA goes and gets associated with the central core of this subunit which is formed around it. Secondly, then the binding of RNA and dissociation of the disc into a lock washer occurs. So, you can see the B part of the figure, that is how this RNA molecules get associated with the protein monomers, and now this protein capsid unit takes the shape of a lock washer and entangles the RNA inside it. So, this keeps on going, and as I showed you in the initial part, 3 nucleotides associate with each protein subunit. So, the 3 nucleotides associate, and by the time one particular helix is formed, and in that turns 49 nucleotides and 16 1/3 proteins are there. So, in the C part you can see that another disc comes in and it keeps on forming the helical structure like this. So, the discs, the capsid discs keep on coming. So, they keep on getting associated with the RNA become a lock washer kind of an arrangement, and the RNA becomes entangled inside this protein capsid protomer. So, that is how the assembly, the self stage assembly of the tobacco mosaic virus takes place. So, we talked about the genome organization, the components of the genome in tobacco mosaic virus, and we also looked at that structurally how this RNA is associated with the, or how it is packaged basically, because there is no nucleus as such here, but how it is packaged or associated with the protein subunits to give rise to the tobacco mosaic virus entirely, which is there. So, now we move on to what is the T4 bacteriophage. Now, bacteriophages are the viruses which can infect bacteria, and T2 bacteriophage or T4 bacteriophage are very important, and we study them a lot in the molecular biology. So, we look at it, how it looks like. So, it contains a linear double stranded DNA. So, it is a DNA virus, one thing. The strand are double stranded and not single stranded. So, it is a linear, and it is a linear molecule, so not a circular molecule. So, it contains a linear double stranded DNA with usually the chemical modification that its cytosine residues. And the genome is again enclosed in a capsid and attached to a tail. So, you can see, and you must have looked at this kind of a structure many a times, that a bacteriophage looks like. It has got a icosahedron kind of a structure on the head, which is the protein capsid which is present. Then it has got a collar, then it has got a core, and then in the core there is a sheath, Then in the sheath it has got tail fibers, there is a tail region which is present. So there is a capsid, and inside this head part, the capsid part, the protein part, the DNA is basically packaged inside. So this is how the bacteriophage looks like. The T4 genome, now the characteristics of the genome, it is 168,903 base pair, and it encodes about 300 gene products. It is an elongated icosahedron, as I just explained. Then the elaborated tail structure is there, including a collar at the base of the head and a rigid tail core surrounded by a contractile sheath. The core and the sheath are attached to a hexagonal base plate. Also attached to the tail plate are tail pins and skinked tail fibers. So this is basically just the structural organization of T4 bacteriophage, much not related to the genome organization, but just to tell you how a bacteriophage looks like. So this is how basically it looked like. And more than 40% of the genetic information is devoted to the synthesis and assembly of the prolate icosahedral heads, tails with contractile sheaths and 6 tail fibers. So 40% of the genome, the genetic information which the genome carries is related, is dedicated to giving this shape to the bacteriophage. So for the synthesis and assembly of the icosahedral heads, tails with contractile sheaths and the tail fibers. That is how basically the genetic information is, and of course the rest 60% can be related to the DNA replication or the transcription part, or basically which will give rise to more particles, and rest of it will depend upon the host machinery. Rest information will come from the host thing. So if we look at the genetic information here, or the genome organization here, then there are 24 proteins are involved in head morphogenesis, and there are 22 for tail morphogenesis and 7 for tail fibers. So these are again just numbers, but as I told you that 40% of the genetic information is involved for the head, the tail structure and the tail fibers or contractile sheath which needs to be formed. Of the 24 proteins assigned for head morphogenesis, 16 are involved in prohead generation and formation and maturation, 5 are responsible for DNA packaging, and 3 complete and stabilize the head. So head forms the most important part of the T4 bacteriophage. It's the important part of the T4 bacteriophage because it contains the genome. And if you remember about the cycle of the way it infects the bacteria, it sits on the bacteria and it just injects the DNA inside the bacteria. So the icosahedron protein remains outside, the tail remain outside, the entire outer structure remains outside, it's only the DNA which is injected inside. And this DNA needs to be packaged inside this icosahedron head. So all these proteins or all these genes which are present in the genome of the T4 bacteriophage give rise to the proteins which aids in that, which aids in the packaging. There are 5 proteins responsible for that, formation of the head and different kind of morphogenesis which is occurring. Only 10 of the 16 genes for prohead formation are essential. So that is also an important aspect, that only 10 of the 16 genes are important. Now, T4 genome is circularly permuted and terminally redundant with respect to base sequence. We will look at now what these two terms mean. The significance of this is that these features protect against the information loss during replication of a linear DNA. Now, as I told you, it is a linear double-stranded DNA molecule. So, the linear double-stranded DNA molecule whenever replicates, then you have already seen the or understood the mechanism of DNA replication. Then DNA replication is a primed mechanism. There has to be a primer. The RNA primer binds and then the DNA is duplicated, or the nucleotides are on the head of the primer. So, the primer needs to bind to some part of the DNA, the complementary part of the DNA, and then the synthesis needs to take place. Now, if it is a linear DNA molecule, then at the end always there will be some portion which is left where the RNA primer cannot bind. It does not have anything else on the upper part where the RNA primer can bind. So, with every round of replication in the linear DNA molecules, with every round of replication, some part of the DNA will not be replicated. So, the one thing is that DNA will be shortened, and the second thing is because that is not replicated, it will remain as a single strand, which is a part of which is basically a source for the DNA damage to occur. So, now they have circumvented this problem by creating these two, by associating these two mechanisms. One is called as the circularly permutation and another is called as the terminally redundant. So, the figure explains what a terminally redundant thing is. If there is a genome, for example, and it has genes say AB, ABCD, CDEF, EFGH in such a manner, then when this genome has to be, when it has been replicated, these are just the segments of DNA which are there. So, always the division or the cutting occurs a little more than the actual genome size which is there. So, the genome starts with AB and then it ends with IJ. So, it is AB, CD, EF, GH, IJ. But this AB, CD, EF, GH, IJ, when it needs to be cut, then the second part, the cutting will occur a little ahead than the HIJ and will include the AB. So, the segments which you get are shown in the lower panel, which is the AB on both the sides. So, it is terminally redundant. So, at terminal portion, the genome sequence is repetitive in nature. So, it is a terminally redundant segments they will give rise to. So, every genome which has been replicated, every part of the genome will have a terminally redundant ends. One has AB, another has CD. So, that is how the terminal redundancy occurs. An important aspect in how this terminal redundancy occurs, one has to understand one mechanism of replication which is called as the rolling circle replication. So, what happens in the case of rolling circle replication is that, for example, if it has a double stranded DNA molecule or if it has a single stranded DNA molecule, as you can see in the figure, then one of the strand is nicked by the restriction by any endonucleases, by a nuclear endonucleases. So, one strand is nicked by an endonuclease. So, there is an open 5' end and a 3' end, and then the free 3' end is extended by DNA polymerase, which reads in the 5', 3' direction, displacing the complementary template strand, which is copied in short segments by the DNA polymerases. So, that circle is transcribed from 5' to 3' end, and as you can see, there is a black arrow which is showing that the blue circle, the blue strand just replicates itself from one direction, and the other part which is extended is replicated in segments, and you can see that the segments are formed. The replication proceeds until at least one new copy is made. The multiple copies may result chained together as a concatemer. So, it should not stop at one point. The replication will keep on continuing, and one genome will not be replicated just once, but it will contain the one set of genome and then again and again repeating the same genome one after the other. So, tandem repeats of the same genome, basically called as the concatemers. So, they will be linked together and then the cutting will occur. So, when the cutting will occur, as I said, this cutting will ensure that the cut portion is a little more extended than the genome. So, it started with AB, ended with IJ, that is ideally one genome segment, but when these are concatemers, then there is a repeat of AB to IJ, then again AB to IJ, then again AB to IJ, all linked together. So, when this cutting enzyme will come, it will cut in the sense that it will cut after first AB and the second AB start. So, at both the ends it has AB which is present, the concept which I just explained about the terminal redundancy. So, terminal redundancy is usually associated with the cyclic permutation and occurs in the case of a rolling circle DNA replication, and therefore, it helps the DNA to be replicated. So, replicates to produce linear double stranded DNA copies with what is called as the sticky ends, because they are terminally redundant. The single stranded regions and concatemers are formed by enzymes joining together several single copies of the genome by zipping together their single stranded sticky ends. So, as I told you, there are sticky ends which are present, so there are concatemers which are formed. So, we were talking about T4 bacteriophage DNA. So, coming back to that, so you can see that now how this DNA is packaged inside the head of the virus or head of the bacteriophage. So, there is a concatemer which is present, which is the repeated sequence of the set of the DNA of the genome. Then the packaging is done by an enzyme which cuts it a little more. So, if you can see that, and every progeny will have a probably different kind of an organization, because every progeny, for example, is giving rise to a different kind of a terminally redundant portion. So, in the first part you have got ABC, ABC, in the second part you have got DEF, DAF. So, all the progenies, even if coming from the same parent, might have different set of organization in the case of viruses, but one, they have a complete set of genome. The terminal redundancy can be different, therefore packaging can be different, but the entire set of genome is present inside the head. So, the packaging of DNA into the head of T4 bacteriophage, you can see the circular genome is represented by, as I said, A, B, C, D, E, F, G, H, and it is repeated in the concatemer, and the circular permutation with terminal redundancy. The packaging of DNA is initiated from the double-stranded ends. For intracellular concatemers, the terminase complex initiates packaging by first generating the ends. So, the enzyme which causes this cuts in the concatemers is the terminase enzymes. Then the terminase DNA complex is translocated from the cytosol to the portal protein at the vertex of the head to form what is called as the packazome. So, the terminase first gets associated with the DNA complex and it translocates from the cytosol to the portal protein which is present at the vertex of the head to form what is called as the packazome, which uses the energy of ATP hydrolysis to translocate the DNA into the head. Now, DNA as such, as a linear stranded DNA molecule, is packaged in such a manner inside the T4 bacteriophage head. The head full of DNA is about 3% longer than the genome size, accounting for the circular permutation of the T4 genomes with terminal redundancy of the each genome. So, each genome has the similar ends and they are circularly permuted when they are basically replicated. So, each DNA, each genome, each head will have will be full of DNA which is about 3% longer because they have repeated ends, and as I said, the cutting is done a little more than the actual genome. So, a little more actual genome is cut, so 3% longer than the normal genome size. And another important point to remember is that the progeny will, all the progeny will have different ends of, they will not have the same ends which are there. So, they might have different organization of the genomes therefore present, but that is how the overall packaging of the DNA or overall organization of the genome is there inside the head of a T4 bacteriophage. So, what we did discuss till now is basically the bacteria, the two kinds of viruses, the TMV and the T4 bacteriophage, that what kind of how the DNA is organized or how the genome. We talk about genome because we are generally talking about the genes here, you know, the transcribed part or the translated part which will give rise to a functional protein. So, we are talking about the genome organization because genome is the complete set of genes which are present. So, the DNA which is making to some extent some sense how it is packaged. So, we have talked about these things. So, now moving on from the prokaryotic to the eukaryotic genome organization. Eukaryotic genomes, you know, are very diverse. The genome of eukaryotes are composed of multiple chromosomes. So, there is not a single chromosome which is present or a single DNA molecule which is present and then organized into chromosomes or packaged inside into the heads of the viruses, but there are multiple chromosomes which are present in the eukaryotic system, and each chromosome contain a linear molecule of DNA. To get the idea about the diversity of the genomes of eukaryotes, you can look at the table. There are organisms, then there is the genome size which is basically in mega base pairs and the chromosome number, and here we talk about the haploid. So, I understand that you know about the haploid set of genes or the diploid part of the organism, which is basically the one set of genome is said to be a haploid set. If they contain two same sets of genomes, then it is said to be diploid and so on. So, this is basically a haploid number, a cell which contains a single set of genome of that particular species. So, for example, in case of yeast Saccharomyces cerevisiae, the chromosome, there are 16 chromosomes which are present and the genome size is about 12 MB. So, you can see the diversity then ranging from yeast to humans. So, humans has approximately 3000, the genome size, there are 3000 MB which is there, and there are 23 chromosomes which are present, but you can see some of them have lungfish and lily, they have huge genomes which is approximately 50,000 MBs, and they are organized into, for example, in lungfish into 17 chromosomes. In case of lily, the 50,000 genome size is organized into 12 chromosomes. So, you can see the diversity that there is a huge sizes which are there for the genome size, and in fact in the chromosome number also there is a lot of variation. Sometimes a smaller genome is organized into so many number of chromosomes. So, that is a characteristic of a particular species, but this table was entirely to tell you about the diversity which is present in the case of eukaryotic system. Now, this chromosome structure changes as the eukaryotic cells divide. So, this is a typical cell cycle which is typical division which is shown here. So, there is an interphase cell, then it enters into, for example, a mitosis is shown, and it undergoes different stages which is prophase, prometaphase, metaphase, anaphase and telophase, and finally the cytokinesis when the cell divides into two. So, you can see that the arrangement of this DNA or the organization of the DNA within the cell is changing with the phases of the division. So, in the interphase, the chromatin, which is called as chromatin, is a little diffused, but as you move towards the metaphase to anaphase transition, then it becomes more and more compact and condensed. So, it becomes the chromosome is more condensed, and finally it gets transmitted. So, one of the very essential important function which we talked about, the organization of DNA into this chromosomes, was that the transmission occurs, the transmission to the daughter cells occurs efficiently. So, this is what is very important here, that it from a very loose or diffused DNA, it tends to become more and more condensed, and tends to become more condensed so that it can be transmitted or divided equally between the two daughter cells which are going to be formed. So, this is mitosis, just a simple mitosis which is shown. So, now we will follow this organization in eukaryotic system. So, we will see that how the DNA is organized into this interphase region. Then we will also talk about the different levels of genome organization from a double helical, single, simple, linear DNA molecule, double stranded DNA molecule. So, how does it become this condensed chromosome of a metaphase stage, or how does it become a condensed chromosome of the anaphase stage which get divided, which goes segregated into the two poles and finally transmitted into the two daughter nuclei. So, we will follow this route in eukaryotic system here. Two important proteins, as I told you, DNA is associated with a lot of proteins which are present there. So, we will talk about the order of structures, but at this point I just wanted to mention these two important structural maintenance proteins, as they are called as, which are called as the cohesins and the condenses. Apart from the different proteins which are associated with the DNA directly, we will talk about them. You know about the histones, the non-histones, etc., which give rise to the higher level of organization. These are the proteins which are the components of the nuclear scaffold. So, in eukaryotic system, you have got a nucleus. That is why it is a eukaryotic system. You have got a well-defined nuclear membrane, and the nuclear matrix is there. The DNA is lying inside the matrix, and this matrix has a lot of proteins which give rise to which basically provides the scaffold. So, this nuclear scaffold has two important proteins, which is cohesin and condensin, and they play a very, very important role in making the DNA how it looks inside the cell. It is not just a double stranded DNA molecule how we saw when we were studying about the polynucleotide chains, but how it is organized inside the cell. These two play an important role. The two, as you can see, one is the cohesin, which basically keeps together the two strands of DNA, and then there are the condenses which keeps together the different loops which are formed. So, these cohesins and condensins, we will talk about the role of these DNA molecules in detail further as we go. So, we will talk about the features of the interphase chromosome. We will start with the interphase chromosome, although we do have to start with from the DNA double helical structure, but I just thought to divide it with the cell division will take place, how the cell division is taking place. So, we will talk about the interphase chromosome first. The light microscope studies in the 1930s distinguished between two types of chromatin. In the interphase nuclei of many higher eukaryotic cells, a very highly condensed form is present, which is called as a heterochromatin. So, you can see this is an electron micrograph of a nucleus, and inside the nucleus you can see there is a darkly stained region which is called as the heterochromatin, and a less condensed region which is called as the euchromatin. So, the heterochromatin and the euchromatin portions are there. This is another electron micrograph in which you can see that the heterochromatin portion is usually present near the periphery of the nucleus. So, you can see there is a nuclear pore, there is a nuclear membrane, and the darker portion is present on the periphery nearer to the nuclear membrane, and the rest of the part is the euchromatin. Euchromatin is composed of the types of chromosomal structures, which is 30 nanometer fibers and loop domains. We will talk in detail about this, that what is a 30 nanometer fibers and loop domains, but for now you should understand that euchromatin is composed of a certain kind of a structural organization which is differing from the organization in case of heterochromatin. Heterochromatin in contrast includes additional proteins and probably represents more compact levels of organizations. So, we just saw the presence of cohescence and condenses. So, they are basically compacting and condensing the chromatin more at these regions which are called as the heterochromatin part of the region. Excuse me. So, if you compare euchromatin versus heterochromatin, the euchromatin stains positively with the DNA specific stain. So, we just talked about the basic fission. I just mentioned this thing, and the reaction is called as the Feulgen reaction, in which the DNA is stained with a particular kind of a stain that indicates a concentration of DNA. The euchromatin is genetically active and stains lightly, and it represents the area of less condensation. Heterochromatin, however, stains more deeply than euchromatin and represents highly condensed regions on the chromosome. It tends to be located at the nuclear periphery, as just we saw, where specific interactions with the envelope may occur and often forms blocks surrounding the nucleolus. It is usually the not transcribing part, although it is not correct, but it's not entirely correct. There are certain portions in the heterochromatin also which are transcribing, but generally it is viewed as a non-transcribing as compared to the euchromatin, which is very transcriptionally active part, and it is usually the late replicating part also, the heterochromatin. So, if you can see the generalities between euchromatin and heterochromatin in the interphase and as it transforms into a metaphase chromosome, the heterochromatin is always tightly coiled, whereas the euchromatin is loosely coiled in the interphase which is present. Then there is two kind of heterochromatin which is there. One is called as the constitutive versus facultative. So, as you can see, constitutive means always tightly coiled. So, constitutive heterochromatin is a permanent feature. The condensation is a permanent feature of a chromosome. For example, we'll look at the part of the chromosome such as the centromeres. So, centromeres are always condensed, or telomeres are always condensed. So, they are constitutive heterochromatin. However, there are certain facultative heterochromatin that they are not always present as heterochromatin. For example, the X chromosome body. So, you must have, there is a female cell and there is a male cell. The female cell contains XX chromosome, the male cell contains XY chromosome, and for the dosage compensation, because now the female cell will have two sets of X chromosomes, one of the X chromosome becomes very much condensed and it is termed as the bar body or the sex chromatin. The bar body after the name of its discoverer. So, that condensation is facultative chromatin, because now when this cell will pass on these two chromosomes to the daughter nuclei, then both of them will become again the euchromatin part. It's not necessarily that it's not there that the one condensed form will always remain condensed, because these two X can be given to one X to each daughter, and then it can be accompanied with a Y chromosome or a X chromosome coming from the other parent. So, this kind of condensation which is reversible is said to be a facultative heterochromatin which is present. So, the constitutive heterochromatin is always compact and tends to be enriched in repetitive, gene poor, because it is less transcribing, and late replicating DNA sequences. However, facultative heterochromatin can reversibly undergo transitions from a compact, transcriptionally inactive state to become more open,
and transcriptionally competent cells. So the DNA. So you can see the difference between that. There is stable and reversible. It contains mostly, we'll talk about this, what a satellite DNA is, but these are the repeated DNA sequences, whereas facultative is usually rich in the long interspersed nucleotide sequences which are there. The polymorphism is plus and minus. The polymorphism means that constitutive heterochromatin can occur in many structural forms, whereas facultative heterochromatin usually have only one structural form when it is condensed or it is uncondensed. Then there is a kind of staining which can occur, which is called as the C staining, done by the Giemsa stain. The C bands is plus in constitutive heterochromatin, that the heterochromatin part takes up this stain, whereas more densely, whereas facultative heterochromatin you are not able to find such kind of an alternate condensed or the non-condensed forms basically. So we'll talk about the properties of heterochromatin. The heterochromatin, as I said, is condensed, it is late replicating, it is methylated. So most of the DNA which is heterochromatin is also methylated. The histones are hypoacetylated. So we'll look at the structure of DNA that they associate with certain proteins which are called as histones, and these histones particularly in the heterochromatin part of the DNA are hypoacetylated. Then histones from heterochromatin are also methylated on lysine 9. The hypoacetylation also occurs on the lysine residues, and they are also methylated on their 9th region of the lysine. Then they are transcriptionally inactive, as I said, because they are more compact and constant. Then they does not participate at all in genetic recombination, and they has a gregarious instinct. So because we will talk about the polytene chromosome present in the salivary glands of Drosophila's larvae, that when the central portion where the chromosomes attach, the centromere, they tend to aggregate. So heterochromatin has an aggregating kind of a behavior which is present. So I think we will continue with this in our next lecture. We'll talk more about the organization of DNA in the eukaryotic chromosome, the different levels of organization, starting from the DNA double helical structure going up to the metaphase chromosome, and we'll also talk about a little more about this facultative heterochromatin and the euchromatin in our subsequent lectures. Thank you.
Thank you very much, ma'am. May I ask you a couple of questions that came to my mind regarding the subgenomic promoter in virus? Can you please elaborate to our students about it?
The particular thing which I talked about was the tobacco mosaic virus. In tobacco mosaic virus, it's a RNA. It's a RNA genome which is present. So RNA genome basically means that it is already in the transcribed form. So this RNA can directly be translated into certain proteins. So half of the certain part, the 5' part of the RNA is basically generally translated into the protein form. But it also contains this subgenomic promoter. So the role of the promoters is basically that the entire machinery will come sit there and transcribe this into another transcript which can then be translated. So promoter is a region where the machinery assembles from where the transcription of the gene can start off. So some of the RNA act as a normal DNA like a thing, it's not a DNA, but DNA like a thing, that it has got a promoter. The transcription machinery will sit there and transcribe into another transcript of RNA, but some of the RNA, because it's a RNA, can directly be there. So those subgenomic promoters are very essential for the transcripts to be formed.
Ma'am, when we were looking at the tobacco mosaic virus and how the genetic material was bound to the protein and it was forming both the structural as well as the genetic aspect, it is very similar to when we see the histone bound system in eukaryotes.
Yes.
So would you comment on it?
See, in tobacco mosaic virus, I would not say that it's very similar, but the association with the proteins is there. And for genome organization, everywhere the association with the protein is there.
Yes.
In tobacco mosaic virus, as such, the RNA is naked. The genome is naked. It directly does not get associated with any of the protein molecules, like in eukaryotic DNA, what we will see, that they do associate with histones and everything. But the packaging in the tobacco mosaic virus is as such that it does interacts with the coat protein. So it appears to be like it's a helical structure, but it is not actually wound around the protein core or anything
as
like
a
we will see in the
nucleosome.
DNA molecule.
Yes, not
Exactly.
like a nucleosome.
It's not like that. It's just because the capsid protein is there. So it intercalates between inside that. Otherwise, the RNA by itself is a naked
molecule.
RNA molecule.
Thank you so much, ma'am. Ma'am has discussed all the aspects about viral DNA in details, and they were greatly fascinating. We will of course be discussing more about heterochromatin and euchromatin, and you will understand more aspects about regulation and epigenetics once we unfold with more lectures on this. So students, if you have any queries and if you have any concepts which you are not able to understand, please do write into us so that ma'am can take them up. And please follow this lecture on YouTube channel and leave in your comments. Thank you for watching. Thank you, ma'am, for this very enlightening talk.
Thank you.
Thank you, ma'am.